much for this invitation. My name is Renzo Ramirez. I am a plant pathology student in the University of Florida. I'm working with Dr. Balat here in the Gulf Coast Research and Education Center. And today I'm going to talk to you about one of the most important diseases in cabbage in the world, the black rot. So in today's presentation, I would like you to, at the end, you will learn about the characteristics of the causal agent of the black rot in brassicas, also the challenges to manage black rot of cabbage in Florida, and at the end, I will give you some information about the research that I'm working with Dr. Balat that is in search of couple alternatives to manage this disease. So to begin with, I would like to mention some information about the brassicas. Brassicas are the third most important group of vegetables that are produced globally, and they have the cabbage as the most important crop within the genus. When we talk about production of cabbage in the United States, Florida is the third largest cabbage producer, and the USDA has registered 9,000 acres of cabbage that produce 125,000 tons of this vegetable, and results in a profit that is very close to $50 million. But just like many other crops, cabbage is also threatened by different diseases, and one of the most important is the black rot. In the case of Florida, black rot reports loses up to 10% of the yield every year. So this disease is caused by a bacteria. The bacteria is Xanthomonas campestris, Pathovar campestris, which I will be referring as XCC. Some important features about the bacteria is that it's a gram-negative and motile. So you can see here in the picture on the left, this was a picture that was taken in an electron microscope, and we can see, give me a second, yeah, here. And you can see here the flagellum that actually helps the bacteria to move. Other important feature is that just like other Xanthomonas species, it typically has carotenoid pigmentation. And this is actually very important at the moment that we're working in the lab, because when we plate the bacteria XCC in a plate, we can recognize it because of these yellow colonies that are going to start growing. Additionally, temperatures around 77 to 86 Fahrenheit and days with fog and rain are very favorable for the development of XCC. Some of the most important symptoms that we can find in cabbage with the black rot, I have divided in two. One is the seedlings and other in the older stages. So as you can see here in the picture above, on the right, you can see a healthy plant that was not inoculated with XCC. And on the left, you see two plants that were inoculated. The characteristic symptom at this stage is the dwarfing and lower leaf drop. In the case of older plants, especially now we're looking at them in the field, we notice that the cabbage has a very characteristic V-shaped chlorothic lesions that are going to appear on the margin of the leaves. But not only that, we also see that this disease can spread into the heads, and internal damage is going to be found because we notice that it's going to be discolorated, and also there's necrotic lesions in the vascular tissue. It is also important to know the disease cycle of the pathogen. We know that XCC is a seed-borne pathogen. This means that it mainly disseminates through seeds. So if we start our season using infected seeds, then we are going to have infected seedlings. However, this is very interesting because most of the time we don't see symptoms in this stage under greenhouse conditions, because first, it's possible that the environment is not favorable for the development symptoms, but also can be because there's a low concentration of bacteria that is not enough to show symptoms at this stage. Other important source of inoculum of the bacteria is that they actually survive in plant debris. There has been research done, and they have concluded that XCC can survive in brassica debris for at least 2 years. And when the environment is conducive, the dispersal by rain and wind, and also the water splash, can take this bacteria to our susceptible host. Once in the host, the bacteria is going to enter the plant through natural openings that are located on the margin of the leaves, which are called the hydatodes. And after that, it's going to start infecting and colonizing the leaf and vascular tissue systemically. Around 14 days after the infection, we're going to look at the development of lesions on the leaf, and afterwards, bacteria process is going to go into the stem and the roots, and we can see xylem decay. Now, knowing about the disease cycle is very important, because knowing that, we can actually target management tools. So, for example, we know that XCC can survive in plant debris. So in order to establish some management tools, we have crop rotations. We know that XCC is not only restricted to cabbage, it can also affect other brassicas like cauliflower, kale, and even brassica weeds. Especially brassica weeds are one big source of inoculum, especially when we have next to our cabbage field this weed, the wheel mustard. Under good conditions, the bacteria that is growing here can just spread into our cabbage field very easily. Additionally, we know that XCC is a seed-borne pathogen, so it's recommended to use certified disease-free seeds and seedling, and also use resistant varieties at the moment that we start our season. Now, we know that at the moment that we start looking symptoms in our field of XCC, the first product that comes to our mind, it might be copper. Copper has been used for many years to manage bacteria diseases. However, in the case of the Florida industry, there is a concern about the appearance of copper-tolerant strains. There has been reports in Florida about the appearance of Xanthomonas citri and Xanthomonas perforans, copper-tolerant strains. And what these strains are going to do is actually do the work of the grower even harder, because they need to elevate the rate of copper that they put in their field. But not only this is a problem for the budget of the grower, it also can lead to environmental effects, because this copper can be accumulated in the soil after constant applications. So we have identified the most important challenges to manage the black rot in Florida. First of all, Florida's environment is ideal for black rot outbreaks. We know that Florida, for most part of the year, is typically humid and warm. So this actually is very favorable for the disease cycle of XCC. Additionally, growers basically relied mostly on copper-based products. And by just using and relying on copper, we're creating a high selective pressure for these copper-tolerant strains that are going to make the management even harder. So taking that in consideration, our main objective is to determine the efficacy of copper alternatives to manage the black rot in cabbage. So what I'm going to start doing now is talking about the research that we are conducting here in the Gulf Coast Research and Education Center. For this set of trials, we always use the cabbage cultivar Bronco. We grow it here in our greenhouse, and then we transplant it into the fields. And we wait in the field until the cabbage is in the pre-cupping stage, as you can see here in the picture. And then we start the application of the treatment 7 to 4 days before the inoculation. After that, we continue the treatments applications weekly for 5 to 6 weeks. The inoculation was done using a backpack sprayer with a bacterial concentration of 5 times 10 to the 7 CFUs per milliliter, and it was performed twice in each trial. In the case of the evaluations began 10 days after the inoculation, and it was we did weekly ratings of the of the plants, looking for the percentage of disease severity in each of them. At the end of the trial, what we did was calculate the area under disease progress curve. This is a summary of disease over time that is going to tell us how much disease we get at the end of the season in each treatment. Finally, we perform a harvest. The harvest was done manually, and each of the head was weighed and cut in the half in order to look for internal black rot symptoms, as you can see here in this picture. So anytime that we look for these symptoms during the harvest, we didn't take that head in consideration to calculate the marketable yield. So today I'm going to present the efficacy test that we performed, and I have separated in two groups of products. First, I'm going to present the sulfur and plant defense activator products that we use. So we started this trial to determine the efficacy of products in alone and mixed application with sulfur, and then we want to compare the results with a grower standard and a not treated control. Other set of trial was we want to use the surfactant as a treatment to determine the effect of weekly application of surfactant in the black rot severity. So we know that XCC likes watering conditions, likes to stay in the leaf when there's free water. So we were thinking that the use of weekly surfactant without any product was actually going to increase the disease severity compared to the other treatments. In all the treatments that I'm going to present, I use COSI 3000 as my grower standard. Its active ingredient is copper. Also, I use plant defense activators like Actigar, Baxiplan, Howler, Lifeguard, and Regalia. Additionally, we add Mancozeb, especially in a mix with copper, because we know that it's one of the products that growers use at the moment of manage bacteria, especially in tomato. And finally, the sulfur-based product, mycothiol. You might be surprised that we're using sulfur to manage bacteria, but because we usually use it to manage fungal diseases. But in this case, we have reports from especially growers that they start adding mycothiol in a lawn application or in tan mix, and they have reported very good management and improvement in the management of the black rot in their fields. So taking those reports in consideration, we want to add this product to our trial to see the efficacy against other products that we have. The second set of trials, we use biological products. And in this case, we want to determine the efficacy of these biological products compared to a grower standard and a not treated control. Additionally to the products that I mentioned before, we also add other plant defense activators like Razox, Stargus, and Theia. And also we add Serenade Opti, which mode of action is more targeting disrupting the cell membrane of the pathogen, and also having an inhibition in the attachment of the pathogen to the leaf. So the first set of products is focusing on sulfur and the plant defense activators, and we're going to focus in these 5 products: Actigar, COSI, Mycothiol, Pencozeb, and Baxiplan. The rates that I'm going to present here and the rates for the upcoming trials were selected looking for the most for the higher the highest rate in the label, but also considering the rates that the growers use in their fields. So during the first trial in the spring, we only focus in 3 products: Actigar, COSI, and Mycothiol. What you're looking here is the area under disease progress curve, the AUDPC, which is the amount of disease over time. And we noticed that Actigar and COSI, both of them significantly reduced the disease severity compared to the not treated control. On the other hand, the sulfur product, Mycothiol, didn't show the same significance reduction when we compare it to the not treated control. When we looked into the yield, we noticed that there's no significant difference between the treatments. However, there is a marginal effect of COSI 3000 improving the yield compared to the other treatments. During the fall of 2021, we used the same products that I mentioned before: Actigar, COSI, and Mycothiol, but we add Baxiplan and we add Pencozeb. Not only that, we also add treatments where we mix microthiol with Actigar, the same microthiol with COSI, and the same with Vaxiplan. We also made a mixture of COSI and Pencosep to see the efficacy of this product. When we compare the products to the not-treated control, only Actigar and COSI, both of them significantly reduce the disease severity. And however, microthiol, once again, we notice that it doesn't have a significant reduction on the disease severity. But this graph not only gives us that information, it also shows us how the mixtures of microthiol with the other products doesn't really improve the efficacy of the alone application. For example, here with microthiol and Actigar, we see that there's no significant difference between both of them. The same happened with a mixture of microthiol and COSI applied alone, and the same happened with Vaxiplan. Once again, we didn't see significant difference in the total yield when we compare the treatments. However, here we start looking for something very interesting. As I mentioned at the beginning, we calculate the marketable yield. The marketable yield here in this graph, you can see it in these blue bars. And the total yield is the orange bar here in the graph. So whenever we find in the harvest plants that have similar symptoms to this picture, we didn't take it in consideration to calculate the marketable yield. And we noticed, and even though there's no significant difference in the total yield and in the marketable yield, we noticed that some products are actually reducing the yield during this season. This is something that we saw in subsequent trials. However, it was not consistent. It looks more that it was very sporadic, because in this case, we noticed that it's COSI, the combination of COSI with Pencosep, microthiol with Vaxiplan that shows reduction. However, in subsequent trials, we noticed that these products actually perform similar when we look for incidence in the head, in internal symptoms of the head. So we are thinking to make more evaluations about it, but we also believe that this reduction is caused more by environmental factors, also of timing, and also is caused by timing of inoculation. So we're gonna continue working with this in the upcoming seasons. So some important points to remember for this first part is that Actigar was very effective alternative to copper to reduce the black rot disease. Also, microthiol showed no beneficial effect reducing disease severity or improving the yield, apply either alone or in tan mix. Also, there was no significant difference among the treatments in total yield or in marketable yield. The second set of trials that we conducted were testing the biological products. In this case, we're using our grower standard, Howler, Regalia, Razux, Serenade Opti, Stargus, and Theia. In this case, we did things a little bit different because we didn't mix all the products. For example, here in the case of Razux, we use an alone application of this product, but we also alternate with Stargus. This means that 1 week we only apply Stargus, and the second one we use Razux. The only case that we did a mixture was when we mix Regalia and COSI, and after that we alternate weekly with Stargus and COSI. As you can see here in this graph of the AUDPC, compared to our not treated control, all the treatments reduce the disease severity. However, once we start comparing the products against our grower standard, the copper product, we see that some of them have similar or even better effect reducing the disease. And this is the case of the alternations of Regalia mixed with COSI and Stargus with COSI, the application of Theia, Race, and Razux. So the points that I want you to remember from here is that all the treatments reduce the disease severity during the fall trial that I just presented. And we also have a second trial that we have conducted, and it was only Howler, the product that significantly reduced the disease severity compared to the not-yielding control and also the grower standard. And once again, in here, there was no significant difference in total yield or in marketable yield. The last set of trials that I'm going to present today is adding the surfactant. We use dynamic, and we select the products which had better results in previous trials. In this case, we focus on individual sprays, but we only have mixtures of COSI with microthiol and Pencocept. And one more time, we noticed that during this season, all the treatments significantly reduced the disease severity compared to the not-yielding control. But once we start comparing with COSI 3000, we found out that there are some products with similar or even better efficacy, like Actigard, Howler, Baxiplan, and Regalia. Something that was very interesting and we were not expecting was find that when we use the surfactant, actually it has the lowest disease severity compared to all the other treatments. In this case, we found significant difference in the total yield compared to the not-yield control. We noticed that products like COSI 3000, Howler, and Regalia significantly increased the yield. And it was very interesting to find that the product Actigard, it doesn't have, even though it has the lowest disease severity among our treatments, it didn't actually improve the yield of the cabbage. So just to remember, in this case, all the treatments reduced disease severity compared to the not-yielding control, but only it was improved in the yield when we use COSI, Howler, and Regalia. So the take-home message that I want to give you after you listen to all these trials is that for the black rot management, we have identified some potential copper alternatives, such as Actigard, Howler, Regalia, Razox, and Theia. But we also have unsatisfactory performance coming from microthiol applied alone or in mixtures, when we only apply Pencocept alone. Also, when we combine the improvement in the yield and the disease control, the best products that we found were COSI 3000, Howler, and Regalia. So our research is still in progress. Actually, we're looking forward for some future work that is going to be conduct trials during transplant production. We want to see the efficacy of some of the products that we use, but in greenhouse conditions. Also, we want to evaluate cabbage varieties for resistance to black rot, and we're working on the characterization of XCC isolates that we have collected in Florida production fields. Just to conclude, I would like to thank my advisor, Dr. Gary Balad, and all the lab members of Balad Lab for all the help through these years. I know that it has been hard, so thank you very much for all the help that you have given to me. And if you have questions, please do not hesitate to contact my advisor, Dr. Balad, or myself. We are going to be very happy to answer all your questions. Thank you.